Okay, so welcome to this next video in which we are discussing the SREBP pathway. Okay, right, so in this video what we're going to actually start discussing is the SREBP pathway itself. So, let's start with what SREBP actually stands for. So, it stands for Sterol Regulatory Element, that's the SRE. So that's for sterol, the S is sterol, the R is regulatory, and then the E is element. And then the BP is binding protein, so it's the sterol regulatory element binding protein. Okay, right, so we're going to begin with discussing the different types of sterol regulatory element binding protein, because there's more than one sterol regulatory element binding protein. Okay, so basically there are two genes for sterol regulatory element binding proteins. There is the sterol regulatory element binding protein 1 gene, okay, and then there is also the sterol regulatory element binding protein 2 gene. Now, there are actually then 3 known sterol regulatory element binding proteins, because this gene can actually give rise to 2 different sterol regulatory element binding proteins. So it gives rise to the sterol regulatory element binding protein 1A, and then also the sterol regulatory element binding protein 1C. So I don't quite know what happened to 1B. But those are the 2 that you can produce from the sterol regulatory element binding protein 1 gene, whereas you can only produce 1 known product from the sterol regulatory element binding protein 2 gene. So how is this feat achieved? How do you get 2 different proteins from the same gene? Well, you might think maybe it's different splice variants, but not this time. No, there is actually a different mechanism underlying the 2 different genes here. In this case, it's different promoter regions. Okay, so let's say that this is the main portion of the gene, okay, here. Upstream of the gene you have promoter regions, okay? So the idea is, in the case of the SREBP1 gene, that you have 2 different promoter regions, and either RNA polymerase can come and bind to this promoter region here, and then start working its way along the coding strand and producing the mRNA from that promoter region, or it can start at this different promoter region up here, and then it can get a very different piece of RNA, and potentially this promoter region could be completely shifted of this promoter region. And what I mean by that is that you could have a frame shifting in the way that the ribosome will then read the protein. Alternatively, it could just be that you're getting a little bit extra protein being made from the longer mRNA. The point is that you have these 2 separate promoter regions which will give rise to different RNAs, which will then potentially be read by the ribosome differently. Okay, so 2 promoter regions. Right, so that gives us promoter regions. Okay, so that gives us 3 possible sterol regulatory element binding proteins to work with. Okay, right, so let's now discuss the structure of these sterol regulatory element binding proteins. And because they all have pretty much the same structure. So, they implant into the membrane of the endoplasmic reticulum. Okay, so here is the nucleus, and then we'll have the endoplasmic reticulum here. So this is the rough endoplasmic reticulum. And basically, in the membrane of the rough endoplasmic reticulum, you will find these sterol regulatory element binding proteins. Okay, so let's say this is the cytoplasmic side. Okay, whilst this is the luminal side. Okay, so this side faces into the lumen of the rough endoplasmic reticulum, whilst this side faces out towards the cytoplasm. And basically the structure of these sterol regulatory element binding proteins is that they have 2 membrane-spanning alpha helices, and then they have 2 important domains in the cytoplasmic portion. Okay, so this one is near the amino terminus, so here's the amino terminus of the protein, and then here is the carboxylic acid terminus here, like so. Okay, right, so this important domain that's at the amino terminus of the protein is basically a transcription factor, and it's a transcription factor within the family of transcription factors called BH, LH, LZ transcription factors, which stands for basic helix or loop helix leucine zipper transcription factor, and I will discuss what that actually means later on when we actually see this acting as a transcription factor, but for now it doesn't matter what it means. So this is the basic helix, and then you've got a loop helix, and then leucine zipper transcription factor. Okay, so it's basically two types of transcription factor all in one. So there are types of transcription factors known as basic helix, loop helix transcription factors, BHLH transcription factors, and there are transcription factors known as leucine zippers, and these are actually both of them, and I'll show you how later on. Okay, but this is the domain that will make that transcription factor. Now, this domain is then a domain which is going to bind to another protein, so we might call this the regulatory domain. Okay, so this is the regulatory domain, and it's going to bind to a domain within a protein which is going to regulate these sterol regulatory element binding proteins. Okay, so when we're discussing this pathway first time through, what we're going to do is imagine that cholesterol level is low within the cell. Okay, so if cholesterol level is low within the cell, the pathway which I'm about to show you is going to happen, and what that was going to result in is it's going to result in the production of more LDL receptors and more HMG-CoA reductase enzymes. More LDL receptors will mean that the amount of cholesterol that you can get into the cytoplasm via the endocytosis of LDL molecules is going to increase. More HMG-CoA reductase is going to lead to a greater synthesis of cholesterol. So both of these things are going to help to bring cholesterol level in the cytoplasm up. Later I'll discuss with you what happens when cholesterol is high and how you turn this pathway off. Okay, so the next protein that we need to discuss is a protein known as SCAP. Okay, and this stands for the sterol regulatory element binding protein. That's the S. So the S stands for SREBP, and then the C stands for cleavage. Okay, and then the A stands for activating, and then the P protein. So this is the sterol regulatory element binding protein, cleavage activating protein, which for short is called SCAP. Okay, now SCAP has 8 membrane-spanning alpha helices, and it too starts its life out in the membrane of the rough endoplasmic reticulum. Okay, right, so here it is. So 1, 2, 3, 4, 5, 6, 7, 8. Those are the 8 membrane-spanning alpha helices. This is the amino terminus of the SCAP protein. Then we have a special domain here before you then end with the carboxylic acid terminus here, and I wish I'd got a little bit more space here. And basically, this special domain here, which I'm colouring in blue, is going to bind to that regulatory domain of the sterol regulatory element binding protein, whichever one it is. At the moment we're just treating them all as the same. They all have the same basic structure. So we're not worrying about the different types at the moment. We'll come back to that later. Okay, they all look like this. Okay, now, when cholesterol is low, the SCAP protein is going to be being produced. It will go into the membrane of the rough endoplasmic reticulum temporarily. It will then bind to a sterol regulatory element binding protein, an SREBP, by this blue domain binding to the green domain, and this complex is now going to be moved to the cis Golgi, basically. So remember, the next intracellular organelle is the cis Golgi here. Okay, and I can't draw all I can't draw all 7 cisternae, so I'll just draw 4 here. So we're going to move this complex from the membrane of the ER into the membrane of the Golgi, basically. And this is going to occur via a COP2 coated vesicle. And basically the machinery for COP2 coated vesicle formation on the membrane of the rough endoplasmic reticulum is going to recognize what's known as loop 6 of the sterol regulatory element binding protein cleavage activating protein. So basically, the loops of this SCAP protein are given names. So this loop here is a loop 1. So we go from the amino terminus, then we've got loop 1, loop 2, loop 3, loop 4, loop 5, and then loop 6 here that I've highlighted in red. And basically loop 6 has a special sequence within it which is going to recognize and be bound to the COP2 components, basically. So let's discuss this special sequence, and then we'll discuss how COP2 vesicles are going to be formed. Okay, right. So let me show the sterol regulatory element binding protein cleavage activating protein again. So remember, it has these 8 membrane spanning alpha helices, like so, and then it's got this special domain which binds to the regulatory domain of the sterol regulatory element binding protein here before its carboxylic acid terminus. And in this loop 6, okay, you'll have a special domain here, which I'm going to colour in green, which is going to be recognized by the COP2 proteins. Okay, now what is this sequence? Well, it's basically a sequence of 6 amino acids, and it's known as the MELADL sequence. Okay, so the Mellado sequence. And again, to be able to read this, you need to know your single letter amino acid codes. So let's draw this sequence out. So the first amino acid that you have within this sequence is a methionine amino acid. So here's the amino terminus here, okay, and then it will be bound to the amino acid prior to it. Then we have the alpha carbon with a hydrogen coming off it, and then we'll draw the carboxylic acid group over here, which will be bound to the amino group of the next amino acid along. And the R group, basically, of a methionine amino acid consists of an ethylene group, so 2 methylene groups joined together, then with a sulfur atom coming off it, and then a methyl group coming off that. Okay, so this is the structure of a methionine amino acid. So M is the single letter amino acid code for methionine. Okay, the next amino acid after methionine is E. Now, E is the single letter amino acid code for glutamic acid, which doesn't seem to make that much sense, but G, the single letter code G, stands for glycine, which comes before glutamic acid, so glutamic acid was given E. Okay, so here's the amino group, here is the alpha carbon with a hydrogen coming off it, here is the carboxylic acid group, and then the R group of a glutamic acid amino acid basically consists of a 3 carbon carboxylic acid group. Okay, so here is the 2 methylene groups, and then on the end you'll have a carboxylic acid group. Now, of course, under usual physiological pH, it will have lost its proton, and therefore will actually be glutamate rather than glutamic acid. So glutamate is the conjugate base of glutamic acid. Glutamic acid, strictly speaking, would be the protonated form. Okay, so this is what E stands for. Okay. The next amino acid along is L. Now, L does not stand for lysine, instead it stands for leucine. Okay, so the structure of leucine is similar to valine, but you have an extra methylene group in. So the R group of leucine, here is a methylene group. Then we have a carbon which has 2 methyl groups coming off, 1, 2, and then it also has a hydrogen coming off like so. And then we've got the carboxylic acid group like so. So that's the 3rd amino acid along. And this is L, and this stands for leucine. Okay, the next amino acid along, A, it's very easy. A is alanine. So alanine just has a very simple R group, which is just a single methyl group coming off the alpha carbon, like so. Okay, so that's the alanine. Then here's the carboxylic acid group. Then we have a D, which is aspartic acid. So here's the amino group of the aspartic acid, here is the hydrogen coming off the alpha carbon, and then aspartic acid is basically a 2 carbon carboxylic acid, so a shorter version of glutamate. Now, of course, again, under physiological pH, it will generally be in the deprotonated form, and therefore will be aspartate rather than aspartic acid. Okay, so then you have the carboxylic acid group coming off here, and then you'll have another leucine attached to that. Okay, so another one of these amino acids here, there. So A is for alanine, D is then for aspartate, because clearly A had been used by alanine. Okay, right, so then just add another L on here, another leucine, and then you've got your Mellado sequence. Okay, so this is going to be recognized by the COP2 machinery. So once the SREBP cleavage activating protein has bound to the SREBP, what will happen is this MELADL sequence will be recognized by COP2 machinery, and we're going to bud off a COP2 coated vesicle. Now I'm going to give you a bit more details on how you bud off a COP2 coated vesicle and the proteins involved in that. Okay, so here is our SCAP bound to our SREBP here. Okay, and then just a reminder of the important domains here. This domain here of the SCAP protein is bound to this regulatory domain of the SREBP protein, and then we have our important transcription factor domain here that's going to be important later. Okay, so this is all happening in the ER membrane at the moment, but now we're about to bud this off from the ER membrane, put it into a little vesicle, a COP2 coated vesicle, which will then go and fuse with the cis-Golgi membrane. Okay, so I want to tell you a little bit more about how COP2 vesicles are budded off the ER membrane. And to do this, basically, you have to go beyond textbook stuff. Most of what we have been talking about, in fact, all of what we've been talking about this morning, well, in this video, is textbook stuff. You can find this in textbooks. Okay, what I'm about to tell you is beyond textbooks. It's not conventional enough yet to be considered being put in a textbook. Okay, so you have to go to a review to find this, and I should reference the review that I took this from. Okay, so this review that I took this from is by Randy Sheckman, who is a giant in the world of vesicle buddings off membranes. Okay, so Randy Sheckman has written a very nice review on COP2 coated vesicles and their budding off, and you should go and look that up. It was the 2012 one. It's a very nice short little review with excellent pictures. Okay, right. You can go and look that up to make sure that I'm recounting it correctly. Right, okay, so this is how COP2 coated vesicles bud off the ER membrane then. Okay, so basically it starts off with a protein known as SAR1. Okay, now SAR1 is a monomeric G protein. Okay, which means that it's a single protein which can act as a G protein. It's not a heterotrimeric G protein. It's not coupled to G protein couple receptors. Instead, it's just a single protein which is considered a G protein. Now, what characteristic do you have to be considered a G protein? Well, basically, G protein itself is short for guanine nucleotide regulatory protein, or guanine nucleotide binding regulatory protein, I think is the full thing. So G is short for guanine nucleotide binding and then regulatory. Okay, so what does this mean? Well, basically, the feature that all G proteins have in common is that they have an off state and an on state. And in the off state, they will have guanosine diphosphate bound to them. Okay, so here is guanosine diphosphate. So this will be the off state. Okay, and in the on state, they will have guanosine triphosphate bound to them. So currently, the SAR1 starts off in the off state. Okay, so let's say this is the ER membrane here. Basically, in the ER membrane, you have another protein which is capable of activating SAR1. Okay, so this protein is SEC12. So SEC12 is what you would consider a guanine nucleotide exchange factor. So SEC12 is a guanine nucleotide exchange factor because it's going to basically take in our monomeric G protein, SAR1. It's going to take that guanosine diphosphate nucleotide off, and it's going to bind on a guanosine triphosphate. Okay, so it's exchanging one guanine nucleotide for another guanine nucleotide. And for short, guanine nucleotide exchange factors are often abbreviated as GEFs. So Sec12 is an example of a GEF. You'll see that terminology very often. It's common terminology. Okay, so what's going to happen is SAR1 is going to be converted from SAR1 GDP to SAR1 GTP. Okay, and I should just stress once again that Sec12 is present only in the membrane of the ER, whereas SAR1 is currently in the cytoplasm. Okay, so this is the luminal side of this membrane here, and this is the cytoplasmic side of this membrane. Okay, right. So what's going to happen is that SAR1 is going to be activated by the Sec12 protein, so it now has guanosine triphosphate bound to it, and then when that happens, SAR1 will change conformation, and it basically sticks out a hydrophobic alpha helical structure, basically. So that's what this is supposed to represent. This little line that I've drawn here, this is an alpha helix with a huge number of hydrophobic residues. Now this hydrophobic alpha helix will implant into the outer leaflet, or the cytoplasmic leaflet of this ER membrane. Okay, and that holds the SAR1 GTP now in the ER membrane. Okay, this SAR1 GTP is now going to bind to another little protein complex, which is then going to bind to the cargo that we want to bud off from the ER membrane. Okay, right, so what is SAR1 going to do next? So let me show this. So if we have the ER membrane again, and we'll stick to the convention that the top is the cytoplasmic side and the bottom is the luminal side, then we have our SAR1 GTP bound here. Now I'm going to drop drawing the GTP because although that's very important, I can show that it's got GTP bound just by having this membrane spanning, well, not quite membrane spanning, but membrane sticking into alpha helix here. Okay, so this shows that it's got the GTP bound, so I'm going to stop having to put that there because that will just clutter everything up. Okay, then what's going to happen is SAR1 is going to bind to a heterodimmer. Okay, so floating around in the cytoplasm, there are heterodimers of two separate proteins. Okay, and these two proteins are Sec23 and then also Sec24. So you have this Sec23, Sec24 heterodimer here. So this is Sec23, Sec24 heterodimer. Okay, and what's going to happen is that Sec23 is going to bind to SAR1 here that is attached to the cytoplasmic side of the ER membrane. So can I fit Sec23 in there? I don't think I can, so I'll just put 23 and 24, but remember they are Sec23 and Sec24. Okay, so I think it would be wise to put a bit of colour on here. So we'll have Sec23 in vivid purple here. We'll have Sec24 in green. Okay, and then we'll have the important SAR1 in orange. Now Sec12 has done its job now. Sec12 its role in life was just to activate the SAR1 to SAR1 GTP. We're not going to be interested in Sec12 anymore. Okay, what's now going to happen is that Sec24 will bind to the cargo. It generally is Sec24 that binds to the cargo. Okay, so in our case, the cargo will be this MELADL region of SCAP, but SCAP itself is bound to SREBP, so this is our cargo that the Sec24 is going to bind to. Okay, there is some evidence that maybe other of these proteins, for instance Sec23 and SAR1, will be important in the binding, and it would be naive to say that they aren't going to be involved, at least indirectly, in the binding. But it is generally felt that Sec24 is the main one which binds to the cargo. Okay, so I'll draw the cargo then in here. So we'll just have this sort of blob representing our SCAP with our SREBP. So we'll have the cargo in turquoise here. Okay, right. And this structure that we've now created here is called it's what's known as a pre-budding complex. So this is all forming on the cytoplasmic side of the ER membrane, this pre-budding complex. Okay, and in turquoise here, this is the cargo. Okay, right. So, what you now need to do to actually make this bud off the ER membrane is you need to attach another protein complex on. And this next protein complex is going to be the Sec13, Sec31 heterotetramer. Okay, so it's going to be made up of 4 separate proteins. Okay, so 2 of these are going to be Sec13 proteins, so we'll let these 2 be Sec13, and then 2 of them are also going to be Sec23. Okay, so this is Sec23 here, and this is Sec13, and again these complexes just exist in the cytoplasm. So this is a Sec13, Sec31 heterotetramer. Hetero means different, tetramer means 4-membered complex, basically. So this is a 4-membered complex. It consists of 4 separate proteins all bound together, and they're all different proteins. That's the heterotetramer. Okay, so one of these will come and bind on top of the Sec23, Sec24, like so. Okay, so here it is, and you've then got 2 Sec13s in the middle, and these, whoops, not 23, that's bad, 31 then at the outside. Okay, so cross that one out, that should be a Sec31 as well. Okay, right, that's made a mess. Right, okay, so let's colour in Sec13 in blue, and then we'll have Sec31 in red. Okay, and now what will happen is that these Sec13, Sec31 heterotetramers, which you've got now on the top of the pre-budding complex, those will polymerize with other complexes like this. So, for instance, in the membrane, if I draw the membrane, you're going to have many of these being formed, okay? And I'm not going to draw multiple of them. Instead, what I'll just draw is something like this to represent this entire structure. Okay, so I'm now demoting this entire structure just to being represented as this pink square here. Okay, you will have multiple of these forming in the ER membrane. Okay, so maybe here's another one, and over here is another one as well. Okay, so I'll colour them all in pink. Basically what's going to happen is these pink squares are going to join together, these complexes are going to join together, and it's the Sec13, Sec31 heterotetramer which is on top of all of the pre-budding complexes that is responsible for this. So this top bit here is going to join with this top bit here, is going to join with this top bit here, so that what you start to get is all of these joining together, and the way that they join together results in the formation of a sphere, basically, or a sphere-like structure. So it's very similar to clathrin. These Sec13, Sec31 heterotetramers polymerize together, and when they polymerize together they make a dome-like structure, so that what you're going to effectively get is something that looks like this. Okay, and these 2 lines, by the way, are meant to represent the 2 layers of the phospholipid by there. Okay, so you'll have one like so, then you'll have it bound to another one here, bound to another one here, and this is getting a little bit difficult to draw, but never mind. Okay, bound to another one here, which is then bound to another one here. Okay, so there'll be multiple of them making up this massive great dome, far more than 5, but this gets the point across. Okay, and they will start to pinch off this vesicle, and as the Sec13, Sec31 polymerizes more and more, it will pinch off the entire vesicle, basically, and this will bud this vesicle off from the ER membrane, basically. So let me get another piece of paper then. Okay, so what you then have done is you have your ER here. This is the rough endoplasmic reticulum. Okay, we have the cis Golgi over here, so let's say this is the first cisterna. Okay, and we have now pinched off this vesicle like so, which has got these the cargo in with these complexes assembled on top of them. So it's a little bit too small to draw it there, but basically what you've got is, if we draw the membrane of the vesicle here, so this is the lumen of the vesicle and this is the cytoplasmic side, basically you've got a structure very similar to what you had in the ER membrane. Okay, so here is our cargo, then we've got our SAR1 here, which is still facing out towards the cytoplasm there. On top of that we've got the Sec23-24 complex here, so this is 23, this is Sec24, and then on top of that we've got this Sec13, Sec31 heterotetramer here. So these are 13, this is 31 if I can squeeze this all in. Okay, this is SAR1 here, and this is our cargo here. Okay, and you've got these all polymerized together. Now, all of these proteins that we have now talked about, SAR1, Sec23, Sec24, Sec13, and Sec31, they are known as the components of, whoops, this should be in capitals, the components of COP2 complex. Okay, so when you've got this vesicle which is covered in these complexes, because remember we polymerized Sec13, Sec31 all over the place, so you've got a vesicle covered in these complexes, that is known as a COP2 coated vesicle. Okay, now what does COP2 stand for? Well, COP stands for coat protein. So the CO here stands for coat, the P stands for protein. So this is coat protein 2. Okay, now the COP2 coated vesicle cannot fuse with the cis Golgi whilst it has all of the COP2 components stuck on top of it. So what then has to happen is you have to get the inactivation of SAR1. Okay, so what will happen is SAR1 is a monomeric G protein. Okay, now monomeric G proteins have an intrinsic capacity to turn themselves off. Remember, in the on state, SAR1 has GTP bound to it. So to turn itself off, it needs to convert that GTP to GDP. Basically, SAR1 will have an intrinsic GTPase. So somewhere within its structure, it will have an enzyme which can hydrolyze that terminal gamma phosphate group off the GTP rather to create GDP, and therefore will turn itself off. When it then turns itself off, this entire complex will disassemble, and all you'll be left with is a vesicle which contains the cargo, which remember is our SCAP proteins bound to our SREBP proteins. Okay, right. Now this vesicle will then go and fuse with the cis Golgi, and that will put our cargo into the membrane of the cis Golgi. Okay, and we'll discuss what will happen to the cargo once it reaches the cis Golgi in the next video.